Cellular reproduction. What is reproduction? Reproduction includes a lot of different things, such as the formation of new organisms, new skin cells replacing old dead ones, growing up, wound healing, new cells replacing damaged or missing ones. When a cell undergoes reproduction, the process is called cell division. The two daughter cells that result from cellular division are genetically identical to one another and to the original parent cell. Before the parent cell splits into two, it first duplicates its chromosomes, which are packages of DNA. During cellular division, each daughter cell receives one identical set of chromosomes from the original parent cell. Three functions of cell division by mitosis include cell replacement, growth by cell division, and asexual reproduction. Asexual reproduction occurs in many single-celled organisms, such as the amoeba. The offspring are genetic replicas of the parent. In other words, they're genetically identical individuals. Many multicellular organisms can reproduce both sexually and asexually. For example, some sea star species can grow new individuals from fragmented pieces. Another example is growing a houseplant from a clipping. The type of cell division responsible for asexual reproduction and for the growth and maintenance of multicellular organisms is called mitosis. Mitosis is used in humans for growth, repair, and replacement of old cells. There are two kinds of cell division that take place in sexually reproducing organisms: mitosis and meiosis. Mitosis for growth and maintenance, and meiosis is for reproduction of a new organism. Sexual reproduction requires fertilization of an egg by a sperm. The reproduction of gametes, which are eggs and sperm, involve a special type of cell division called meiosis. Meiosis occurs only in reproductive organs. The gametes have only half as many chromosomes as the parent cell that gave rise to it. Each eukaryotic chromosome contains one very long DNA molecule, typically bearing thousands of genes. The number of chromosomes in a eukaryotic species depends on the species. Chromosomes are made up of a material called chromatin. Chromatin fibers are composed of roughly equal amounts of DNA along with protein molecules. These protein molecules help to organize the chromatin and control the activity of the genes. During interphase, a cell in your stomach lining might make and release enzyme molecules that aid in digestion. While in interphase, a cell roughly doubles everything in its cytoplasm. It increases the supply of proteins, increases the number of many of its organelles, such as the mitochondria and ribosomes, and it grows in size. Typically, interphase lasts for at least 90% of the entire cell cycle. Most of the time, chromosomes exist as thin chromatin fibers that are too thin to be seen using a light microscope. But as a cell prepares to divide, its chromatin fibers coil up, forming compact visible chromosomes that can be seen under a light microscope. Each chromosome of DNA is packaged into an elaborate, multi-level system of coiling and folding around proteins called histones. Histones attach to the DNA. The DNA coils around these histones, forming a nucleosome. The structure of chromosomes varies depending on the function the chromosomes are performing at the time. These are loose and unwound when the cell is undergoing its normal functioning, and they become compact and tightly wound when the cell is dividing. Before a cell begins the division process, it must duplicate all of its chromosomes. After DNA replication, each chromosome consists of two identical copies called sister chromatids, which contain the same genes. These two sister chromatids are joined together at a structure called the centromere. When the cell divides, these sister chromatids of a duplicated chromosome will separate from each other, forming two separate but identical chromosomes. One of the new chromosomes goes to one daughter cell, and the other goes to the other daughter cell. Each daughter cell receives a complete and identical set of chromosomes. The cell cycle extends from the time a cell is first formed from a dividing parent cell until its own division into two cells. You can think of the cell cycle as the lifetime of a cell, from birth to its own reproduction. Most of the cell cycle is spent in interphase. Interphase is a time when a cell goes about its usual business, performing its normal functions within the organism. While in interphase, a cell roughly doubles everything in its cytoplasm. It increases the supply of proteins. It increases the number of many of its organelles, like mitochondria and ribosomes, and it grows in size. Interphase can be divided into three subphases: the G1 phase, or first gap, the S phase, or synthesis phase, where DNA replication takes place, and the G2 phase, or second gap phase. The chromosomes are duplicated during the S phase. At the beginning of the S phase, each chromosome is in a single and linear form. At the end of the S phase, after DNA replication occurs, these chromosomes are doubled, each consisting of two sister chromatids joined along their lengths. These chromatids appear as an X. During the G2 phase, the cell completes its preparations for cell division. The part of the cell cycle when the cell is actually dividing is called mitosis, sometimes referred to as the mitotic phase or M phase. Mitosis includes two overlapping stages, mitosis and cytokinesis. In mitosis, the nucleus and its contents, most importantly the duplicated chromosomes, divide and are evenly distributed, forming two daughter nuclei. Cytokinesis is the division of the cytoplasm into two cells. This process usually begins during the telophase portion of mitosis. In animal cells, the cytokinesis process is known as cleavage. The first sign of cleavage is the appearance of an indentation called a cleavage furrow. A ring of microfilaments in the cytoplasm just under the plasma membrane contracts, deepening the furrow and pinching the parent cell into two separate daughter cells. Mitosis begins in prophase. During prophase, changes occur in both the nucleus and the cytoplasm. In the nucleus, the chromatin fibers coil so that the chromosomes become thick enough to be seen individually with a light microscope. Each chromosome here exists as two identical sister chromatids joined together at the centromere. In the cytoplasm, the mitotic spindle begins to form. Late in prophase, the nuclear envelope breaks into pieces. The spindle tracks attach to the centromeres of the chromosomes and move the chromosomes towards the center of the cell. In metaphase, the mitotic spindle is now fully formed. The centromeres of all the chromosomes line up between the two poles of the spindle. For each chromosome, the tracks of the mitotic spindle are attached to the two sister chromatids, and they get pulled toward opposite poles. This tug of war keeps the chromosomes in the middle of the cell. Anaphase begins when the sister chromatids of each chromosome separate. Each is now considered a full-fledged chromosome. The chromosomes move towards opposite poles of the cell as the spindle tracks shorten. The tracks not attached to the chromosomes lengthen, pushing the poles of the cell further apart, thereby elongating the cell. Telophase begins when the two groups of chromosomes have reached opposite ends of the cell. The nuclear envelopes form. The chromosomes uncoil and the spindle disappears. Mitosis, the division of one nucleus into two genetically identical daughter nuclei, is now finished. Cytokinesis is the division of the cytoplasm. This usually occurs within telophase. The events of the cell cycle are directly controlled by a cell cycle control system. This consists of specialized proteins within the cell. These proteins send stop and go signals at certain key points of the cell cycle. Cancer cells do not respond normally to the cell cycle control system. Instead, they divide excessively and may invade other tissues of the body. In severe cases, they may continue to divide until they kill the host. Cancer cells are thus referred to as immortal cells because, unlike other human cells, they will never cease division. In fact, thousands of laboratories around the world today use a laboratory strain of human cells that were originally obtained from a woman named Henrietta Lacks, who died of cervical cancer in 1951. The abnormal behavior of cancer cells begins when a single cell undergoes genetic mutations in one or more genes that encodes for proteins in the cell cycle control system. These changes cause the cell to grow abnormally. The immune system generally recognizes and destroys such cells. However, if the cell evades destruction, it may proliferate, forming a tumor, which is an abnormally growing mass of body cells. If these abnormal cells remain at the original site, the lump is called a benign tumor. Benign tumors can cause problems if they grow large and disrupt certain organs such as the brain, but often they can be completely removed by surgery and are rarely deadly. On the other hand, a malignant tumor is one that has the potential to spread into neighboring tissues and even into other parts of the body, forming new tumors. A person with a malignant tumor is said to have cancer. The spread of cancer cells beyond their original site is called metastasis. The cells are said to metastasize. Cancers are named according to where they originate. Liver cancer, for example, always begins in liver tissue, even though they may metastasize and form tumors in other areas. There are three main types of cancer treatment: surgery, radiation therapy, and chemotherapy. Surgery is a process to remove the tumor and is usually the first step. Radiation therapy exposes cancerous tumors to high-energy radiation, which harms the cancer cells more than the normal cells surrounding it. Chemotherapy is the use of drugs that disrupt cellular division of cancer cells and are used to treat tumors that spread throughout the body. A typical human body cell, called a somatic cell, has 46 chromosomes. A technician can break open a human cell in the metaphase of mitosis and stain the chromosome with dyes. Pictures are then taken with the aid of a microscope, and these chromosomes can be arranged in matching pairs by size. The resulting display is called a karyotype. Notice in the figure that each chromosome is duplicated, with two sister chromatids joined along their length. Within the white box, for example, each stick is actually a pair of sister chromatids stuck together at the centromere. Notice that almost every chromosome has a twin that resembles it in length and centromere position. In the figure, the white box surrounds one set of twin chromosomes. The two chromosomes of such a matching pair are called homologous chromosomes. Homologous chromosomes carry genes controlling the same inherited characteristics. The 23rd pair of chromosomes are called sex chromosomes. Males have one X chromosome and one Y chromosome for the 23rd pair, whereas females have two X chromosomes for the 23rd pair. The remaining 22 pairs of chromosomes that are found in both males and females are called autosomes. For both autosomes and sex chromosomes, You inherited one set of chromosomes from your mother and the other set of chromosomes from your father. The life cycle of a multicellular organism is the sequence of generation-to-generation stages from fertilization to the production of its own offspring. Having two sets of chromosomes, one inherited from each parent, is a key factor in the life cycle of humans and all other species that reproduce sexually. Humans are diploid organisms because all typical body cells, or somatic cells, contain pairs of homologous chromosomes. The total number of chromosomes in humans is 46. The diploid number is abbreviated as 2N. Gametes are egg and sperm cells. Gametes are made by a process called meiosis, which occurs in the gonads. Gonads are ovaries or testes. Gametes are haploid with an N number of 23. A cell with a single chromosome set is considered a haploid cell. Haploid cells have only one member of each pair of homologous chromosomes. Each gamete has a single set of chromosomes, 22 autosomes, plus one sex chromosome, either an X or a Y. For humans, the haploid number is 23. In the human life cycle, a haploid sperm fuses with a haploid egg in a process called fertilization. The resulting fertilized egg, called a zygote, is considered diploid. A zygote has two sets of chromosomes, one set from each parent. The life cycle is completed as the sexually mature adult develops from the zygote. Mitotic cell division ensures that all somatic cells of the body receive a copy of all of the zygote's 46 chromosomes. Producing haploid gametes by meiosis keeps the chromosome number from doubling in every generation. In step 1, each of the chromosomes is duplicated during interphase. Step 2, the first division, meiosis 1, segregates the two chromosomes of the homologous pair, packaging them in separate haploid daughter cells. In step 3, meiosis 2 separates sister chromatids. Each of the 4 daughter cells is haploid and contains only a single chromosome from the pair of homologous chromosomes. The second difference between meiosis and mitosis is the exchange of genetic material between homologous chromosomes. This exchange, called crossing over, occurs during the first prophase of meiosis 1. As with mitosis, meiosis is preceded by interphase, during which chromosomes duplicate. Each chromosome then consists of 2 identical sister chromatids. The chromosomes consist of uncondensed chromatin fibers during interphase. In the prophase 1 phase of meiosis 1, the chromosomes coil up. Special proteins cause the homologous chromosomes to stick together in pairs. The resulting structure has 4 chromatids. Within each set, chromatids of the homologous chromosomes exchange corresponding segments, swapping genetic material. This process is called crossing over. Crossing over rearranges genetic information. As prophase 1 continues, the chromosomes coil up further and a spindle forms. Homologous chromosomes are moved towards the center of the cell. At metaphase 1, homologous pairs are aligned in the middle of the cell. The sister chromatids of each chromosome are still attached to their centromeres. These centromeres are then anchored to the spindle tracts. For each chromosome pair, the spindle tracts attach to only one homologous chromosome from one pole of the cell. The tracts attach to the other chromosome from the opposite side of the cell. With this arrangement, the homologous chromosomes are poised to move towards opposite poles of the cell. In anaphase 1, the attachment between the homologous chromosomes of each pair breaks, and the chromosomes now migrate towards the poles of the cell. In contrast to mitosis, the sister chromatids migrate as a pair instead of splitting up. They are not separated from each other, but are only separated from their homologous chromosome partners. In telophase 1 and cytokinesis, chromosomes arrive at the poles of the cell. When they finish their journey, each pole has a haploid chromosome set, although each chromosome is still in a duplicated form. Cytokinesis occurs along with telophase 1, and 2 haploid daughter cells are formed. Next, both daughter cells will undergo the process of meiosis II. Meiosis II begins with the process of prophase II. Meiosis II is essentially the same as mitosis. The important difference is that meiosis II starts with a haploid cell that has not undergone chromosome duplication during the preceding interphase. During prophase II, a spindle forms and moves the chromosomes towards the middle of the cell. During metaphase II, these chromosomes are aligned as they are in mitosis, with the tracks attached to sister chromatids of each chromosome coming from opposite poles. In anaphase II, the centromeres of sister chromatids separate, and the sister chromatids of each pair move towards opposite poles of the cell. In telophase II, nuclei form at the poles, and cytokinesis occurs at the same time, resulting in 4 haploid daughter cells, each with single chromosomes. The arrangement of chromosomes at metaphase I determines which chromosomes will be packaged together in the haploid gametes. This is called independent assortment. Independent assortment contribute to genetic variability of the gametes. When aligned during metaphase, the side-by-side orientation of each homologous pair of chromosomes is a matter of chance. In this example, there are 2 possible ways that the chromosome pairs can align during metaphase I. In possibility 1, the chromosome pairs are oriented with both red chromosomes on the same side. In possibility 2, the chromosome pairs are oriented differently, blue-red and red-blue. This arrangement produces gametes with 1 red and 1 blue chromosome. For a human with a haploid number of n equals 23, there are 2 to the 23rd power of possible chromosome combinations. A human egg has about 8 million possibilities, and a human sperm also has 8 million possibilities. So a single man and a single woman can produce zygotes with 64 trillion different combinations of chromosomes. In addition to independent assortment, the process of crossing over also contributes to genetic diversity in the gametes. Crossing over is the exchange of corresponding segments between non-sister chromatids of homologous chromosomes. Crossing over occurs during prophase I of meiosis. At the time the crossing over begins, homologous chromosomes are closely paired all along their lengths with a precise gene-by-gene alignment. These chromosomes are called recombinant after crossing over occurs because they result from genetic recombination. Occasionally, accidents occur during meiosis that can alter chromosome number. For example, an abnormal gamete produced by non-disjunction unites with a normal gamete during fertilization. Here we see examples of non-disjunction in meiosis I and non-disjunction in meiosis II. In both situations, we see abnormal gametes can be produced. When a normal sperm fuses with an egg cell with an extra chromosome, the result is a zygote with a total of 2n+1 chromosomes. Because mitosis duplicates chromosomes as they are, the abnormality is passed on to all embryonic cells. The karyotype shown here has 3 number 21 chromosomes. The person is triploid for chromosome 21 instead of having the usual diploid condition. This condition is called Down syndrome or trisomy 21. A person with Down syndrome has 47 chromosomes. Down syndrome affects about 1 in every 850 children. It is the most common chromosomal abnormality and is the most common birth defect in the United States. Down syndrome includes certain characteristic features, including a fold of skin at the inner corner of the eyes, a rounded face, a flattened nose, short stature, heart defects, developmental delays, a shortened lifespan, and susceptibility to leukemia and Alzheimer's disease. The risk of Down syndrome increases with the age of the mother. For mothers of age 40, the risk of having a child with Down syndrome is about 1%. Fetuses of pregnant women aged 35 or older are candidates for chromosomal prenatal screenings. Non-disjunction and meiosis can also lead to abnormal numbers of sex chromosomes X and Y. Unusual numbers of sex chromosomes seems to upset the genetic balance much less than unusual numbers of autosomes. This may be because the Y chromosome is very small and carries relatively few genes. Males with Klinefelter syndrome have inherited 2 X's and 1 Y chromosomes for their 23rd chromosome. People with Klinefelter syndrome tend to be sterile. They have underdeveloped testes and may have secondary female characteristics such as small breasts. In cases in which a female only inherits 1 X chromosome instead of the normal 2, Turner syndrome develops. In Turner syndrome, the symptoms include sterility and immature sex organs. Mammalian cells normally operate with only 1 functioning X chromosome because other copies of the chromosome become inactivated in each cell. Thank you for watching.